All right. Welcome to the Mur Ligase meeting on 14th of June, 2022. I'm on a single screen, so I'm going to share screen, even though sometimes it's a bit of a disaster if you haven't got double screen, but let's see how we go. Okay, so there's an issue here, number 76, which hopefully has everything. And I guess there's two main things today. There'll be lots of little bits, but two main things. One is that I think we may have some data incoming from the screening of enamine compounds from Joe and Co in Warwick that's been screened by Chris and Adrian. So I think we're going to be talking about that. And secondly, I guess I just wanted to go over the headlines from the SOCIN meeting that we had the other day, where we talked about structural biology and crystallography, and just to sort of highlight what we think is going to be the important next steps for that. After we've talked about those two things, and of course we can talk about anything else. But I mean, I guess my intention was to hand over to anyone in the Warwick team to talk about either of those two things. Either Chris, Adrian talk about data, or Laura to talk about, SOCIN conclusions.
Should we start with Laura, because I'm chasing Adrian at the moment.
So Adrian replied to me. He said he's waiting to join.
Maybe in the Teams meeting. We may have a Teams Zoom problem because the invite may have appeared to be from Teams.
Yeah.
Shouldn't, but
I'm asking him.
It's happened before.
Yeah, it happened to me before as well. It's really easy to click Teams.
Yeah.
I'm sending him the link to Zoom.
No one uses Skype anymore, huh?
No.
Yeah, I still use it with my family. But so if you want, I can go through the structure.
Laura, I think the best thing to do would be to run and find out. He's in another room. He's about 2 meters from you, isn't he?
I don't think he's here.
Isn't he?
I haven't seen him today.
I'm going to phone him. Do you want to start on the structural stuff while I'll chase him up?
Yes.
Okay, I can keep this up here and do this, or if you've got something you wanted to share, Laura, we can do that.
No, it's the same as the slides that I could share the slides from last time, from the other day.
I'm trying to get at the what it is, because I mean one of the really key conclusions was this idea about including UMA in the structures, right, to make them easier to do and to get. So things like that, the sort of take homes for people who weren't here.
So I can do a quick update for the rest of the people that didn't join the structural meeting. So basically, we have been doing Mur E and Mur D from E. coli. We've been working with them for XChem efforts, and the structures were mainly APO structures with no ligands bound, which meant we get open structures. And the ATP sites, the one we want to target to do the dual inhibition or multiple inhibition, is not completely formed. So we're not getting a massive amount of hits. The hit rate is really low. So we think we need to move forward into different conformations of these proteins. So to do that, I ran our different experiments to do soaking to do co-crystallization in the presence of different ligands, and then we could soak into those structures with ligands. And that produced 3 structures that we can use for Mur D, one with ADP, one with AMPCP, and one with the UMA, the monopeptide. So, and we can use that. The one that seems to be more promising because the ATP site is completely empty is the monopeptide structure. So that one is the first priority to follow up and trying to soak compounds into it. And Jan also made progress and got a structure with a monopeptide as well in Pseudomonas Mur D. And he's working to try to get extra things and extra monopeptide structures as well. I'm sending him, hopefully tomorrow, if everything goes well, I'm sending him more monopeptide, and then he can continue with the rest of the experiments. So that's basically what we are in the structural part. And now, yeah, Adrian has joined.
Oh, Laurie, just might mention that you also have some compounds to.
Oh yeah, of course. So I already ordered some compounds from the assays that were inhibiting or that are inhibiting multiple targets. So I ordered them, they arrived yesterday afternoon. So I'm setting up plates this week in different targets, CD and CDE.
And those are the compounds that, those are related to the ones we're about to hear about?
Yes, exactly. That's why I didn't mention before.
And Jan said that later on this meeting, when he's not on the ferry, he's going to update us on a Mur D structure, which I think he's gotten from the Acinetobacter and Pseudomonas, right?
Yeah, exciting. Yeah.
So Laura, so you mentioned, so you're doing something with Mur C as well? Mur C?
Yes. Yeah, I'm purifying Mur C this week.
Okay, that's awesome. That's awesome.
Yeah,
Thank you.
so I will try to do hopefully on Friday. If not, it will be on Monday. Depends on how everything goes. Yeah. That one would be from Pseudomonas Mur C.
Okay. Just before we go to Adrian, two quick updates from us while I'm thinking about it. Ed, you were finishing up purification of the putative dual inhibitors that you and Kato have been working on. And I think are you nearly ready to ship those molecules to Warwick?
Yes. Let me just show this.
So these are fragment elaboration compounds, right? The dual targeting things that we've been previously.
Yeah, so these top 14 compounds should all be ready. So hopefully I'll be able to send them end of this week, beginning of next week. And then, yeah, there's a bunch of these competition compounds, which we're working on now. I don't think I'll have them ready by the time for this first shipment, but probably in a few weeks, maybe.
Okay, that's okay. Could you send me an email 1 week in advance so I can start booking things?
Yeah.
Thank you.
Yeah, so a few teams have suggested molecules already, and I think we're kind of expecting maybe 1 or 2 more teams to come up with molecules. Provided we spend any money we need to spend by the end of the month, we'll be okay, because that's when the grant is over. So we're incentivized to spend by then, and then we will try and make as many of these things or buy as many of these things as we possibly can. All right, great. Thanks, Ed. And then the other thing was, so Yuhang can't join us today. He's got a conflicting thing. He is very, very close to having the original AZ compound. You know, he's been working on changing the OH to NH2 to try and improve its ability to target wild type bacteria. So the primary NH2 is meant to be, you know, helping to accumulate the compound in bacteria. He's very close to making that. We've got a trace that we think is the right molecule with the right mass, and it's pure. And he's just trying to get an NMR of it right now. But with that methodology, he should be able to then make a few other compounds too, which hopefully will hit Mur C in vitro, and hopefully will accumulate better than the original AZ compounds. So he's very, very nearly there. All right. Great. So Adrian, Chris, did you want to present the new data?
Yeah, Adrian, have you got your slides there to share? You made
Yeah.
it last night.
If you, I'm not sure you're going to be able to see them because I can't see you.
What
I'm going to have to do, I'm basically going to have to send them to you and go next slide, something like that.
we can do that.
Yeah,
Yeah.
that's fine.
All right, give me a second.
Convert them into acetate slides and put them in the mail.
You know, I've just thrown away all of my lecture series. I had a whole lecture series on acetates, and I've kept them. I've only thrown them away about 2 weeks ago.
I've still got the acetates I made for my PhD Viva presentation. I don't know.
Chris, you should have melted those things down and made some kind of sculpture you could have put in your desk or something. They use it, they do all that, what's that now, the 3D printing, right? You could use them as input for your 3D printer.
Yeah.
Make a Mur Ligase 3D structure. Sorry.
With a compound.
Yeah, that's right.
How are we doing? Let's have a look.
Yeah.
Have you mailed, Adrian?
Getting there.
Should we come back to this?
I think that's a very good idea. Just give me a second. Yeah, do come back to it.
Yeah, we'll keep going on with the tech. Matt, if you want to take over.
All right. Wait. Okay. Let me just see if there's anything else that comes up here, because I think those are the main things, and we're waiting on.
And Jan needs to get to work.
Yeah, so a bunch of these things were all done. The Atomwise thing, I've not been back in touch with Atomwise more recently. I guess there was one thing there, right, about trying to figure out whether the Atomwise compounds were, wait, there was one action here. Oh no, we've already established that. I think there was a question about whether the compounds were binding by SPR in the presence and absence of ADP, AMP, PMP. Was that still live, Laura?
So I was going to do more essays once we got the compounds from the competition, so I can check all that.
Okay. And
I
think we
did a
didn't.
conversation. There was a conversation about some compounds from the fragment library, right?
So, well, for just the Atomwise compounds, we weren't sure whether or not these compounds were enzymatically active. So they were inhibiting.
So, from what I could gather from Becca's notes, it might be compounds that interfered with the assay. So they were tested, but then no data can be extracted from them. That's what I understood. But Becca is away at the moment, so we will have to wait for a confirmation when she comes back.
All right, be good to know if if they're hitting any of the Murs, obviously.
Yeah.
Just while we've got them, we should try and exhaust all the experiments that we can.
Yeah.
And then we've mentioned the competition, and we're in there from Jan. There was the Merck Open Global Health Library. Chris, did you have any progress on that?
I need to chase them again. They've had. Well, I feel, and them being Warwick contracting. It's the same old thing. Is stuck in our contracting black hole.
Okay. All right.
Yeah. Right. I think it just reinforces for ongoing and future things about trying to get agreed sets of templates signed up by all partners beforehand, because so I don't know, every single I've had to do it with LifeArc compounds as well. That's now all being contracted and signed up, but that hours and hours and hours of work I found for me to go through the contracts and try and persuade people why we just need to sign it off. Ditto every other one as well. So everything that comes through. Okay, I've got Adrian's slides through now.
So we're good to go. Just one last thing before we go on. There was this CC4 carb scheme that we'd mentioned previously. I know Joe and Laurie are familiar with that, which offers hit to lead. I guess I just want to keep that on people's radar in case we do come up with new hits in the near term where this scheme might be suitable. I don't know if Joe or Laurie or anybody else has looked into that in some detail and can advise on whether we might be suitable for it, or are we too early?
I can check.
So definitely not too early. I know Ann Aiken from NIH, who's kind of leading up part of that effort, and XAZ.
Oh, really?
So, I think this. I'm not sure. where they are with, proposals and progress and so on. But I think exactly these kinds of hits, if they get confirmed and we have structure, I think it'd be perfect opportunity. So I think once, we hear from Adrian and maybe, if we get some with or without structure, we obviously want structure, but I think this is something we should be thinking, we should talk about soon about maybe submitting if, in addition to the grant proposals and so on. But anyway, I think it's definitely something we should follow up on.
Okay, great, good.
Cool. I'll have a go at sharing the screen then, and
floor is yours.
Adrian can direct me.
Just a word on the CC4 carb thing, if you don't mind. I mean, it looks like it's an NIAID program, right? So we might be able to assist with that since we're a NIAID program as well. We're supposed to work with them. So if you want a contact person or something, I'm sure our program officer can help.
That'd be useful. Yes.
Does that mean you could come back in again after the end of your term? I mean, I'm thinking for the rest of the year, or does that mean we could continue working with you, Bart, after the or not, or for the rest of the year, if that links up with NIAID?
Yeah.
Oh, brilliant.
We're separate, but there's separate funding and all that, but we're pretty well connected with the NIAID program officers that want us to interact with all their other NIAID programs to leverage their own work. So I don't know. If you're interested in that, I don't know anything about it, but I'd be happy to see what I can help with.
Cool. Great. Okay.
Bart, I'll catch up with you offline about that and how we can kind of tag team from both our ends on that.
Brilliant. Cool. Okay. Adrian, you good to go? I've got my finger on the pointer, so it's over to you.
Okay. Right. So this is very much a group effort over the past couple of months. Two names that haven't appeared before are Christopher Holes and Kieran Duff. They are two final year students who've been working extremely hard over the past few weeks on some of the compounds that we now have an interest in. So if I can go to the next slide. Thank you. So, or not.
Oh, not. Why did that crash? I'm just going to come out of that and go into another window. Dear, oh dear. It was so promising, so poor. We'll try again. We ran into a problem, you're telling me. Right, I'll leave it like this. Okay, there's your slide.
So essentially, the starting point of all of this was a screen of an enamine library targeting ATP binding sites in Streptococcus aerogalacti MURD, which was a project that was initiated between Joe and Swain Backer. And this returned a number of hits, which are actually shown on this histogram of remaining activities. The work was done such that we tailored the assays to be sensitive to ATP inhibition by using an ATP analog, which was deoxy-ATP. And what we then did is that we went on to look at the impact of these compounds on Pseudomonas aeruginosa, MURD and MUR-E. Next slide.
Oh dear, oh dear. I think this is, I think we're blighted today. I have no idea what's happening.
Can you open it in, I think you're in a browser, so can you open it in PowerPoint itself?
There we go.
Maybe try to download it.
The problem is the data is so great that it doesn't want to present it, right?
Shall I send it to Laura and see if we have better luck?
Yeah, let's try that.
Okay, hang on. Right.
Oh, I know what happened, Adrian. I was too previous in clicking open and the whole file hadn't opened.
Okay.
So I think
If
I
you,
know what's
yeah.
happened.
If you can put it in the
Oh, yeah.
desktop version of PowerPoint, I bet that will work.
Yeah, that's where we are now. Can we see that?
Okay.
Cool. Okay.
All right. So we've covered the first two slides. And now, number 3. It always
Chris, can you go into PowerPoint? Can you go into full screen mode, please? Thank you.
maybe. Okay. So the starting point for looking at Mur-D and Mur-E was to establish what the kinetics with respect to ATP were. And in both cases, there were hyperbolic relationships between ATP concentration and velocity, and that gave us good estimates of the kinetic constants for ATP, for Km and Kcat. And I want you to remember the fact that these are hyperbolic, because this isn't always the case, and this becomes quite relevant later on. But anyway, with the kinetic constants we got, we elected to drop the ATP concentration to 1/3 of Km, and then we screened, because it's against the allyl-lactide enzyme, against Mur-D and Mur-E. Next slide. And the result of that was that we got some inhibitors which were dual targeting, for example, LO6, and some which were about 70% remaining at percentage inhibition threshold, which are FO9, N15, and MO2. Next slide. Not the assay that we used, which is an amplex red-based assay, wasn't compatible with all the molecules that Becker had screened. And one of these is JO6, which is pictured, which at high concentrations, millimolar concentrations, required us to use methylguanosine as an assay chromophore. When we did this, it turned out to be a good inhibitor against both Mur-D and Mur-E. Next slide, please. So by the end of the last meeting, we'd started generating IC50 data. So this is for HIT-FO9 from plate A01 with an IC50 of about 130 micromolar and a hyperbolic response. And we then plowed through the other hits that we got, always cognizant that we needed a positive control, which is on the right-hand side, which is a standard ADPCP inhibitor, which gives an IC50 of 25 micromolar. If you go to the next slide. Similarly, we started looking at molecules like LO6. Again, this was somewhat less potent, but we could still measure an MIC. And next slide. And then we got onto molecules like JO6. So JO6 has a 21 micromolar IC50, which corresponds to a 16.6 micromolar Ki, which meant it was actually binding more tightly than the actual standard ATP analog that we'll be using. If you carry on to the next slide. So all that's been with Mur-D thus far. With JO6 with Mur-E, the IC50 was equivalent. Again, it was quite tight. It wasn't quite as tight as ADPCP this time, but still with a respectable IC50 of 16 micromolar, and suggesting a Ki, if it is competitive, of 12.4 micromolar. Now, can we go to the next slide, please? And this basically is JO6 dual targeting both Pseudomonas Mur-E and Mur-D with roughly equivalent potency. And if we could go to the next slide. So the other Mur-E Mur-E hits that we looked at with Pseudomonas aeruginosa were AO2. Again, this is a very, very high IC50 and a very high KI. And the data does show some scatter, and we would probably want to revisit that. But anyway, we could measure IC50s in the hits that were generated in the original Allogalacti screen. Next slide, please. And again, so MO2, which was a hit against aeruginosa Mur-E, has, again, a respectable IC50 for a fragment of 103 micromolar, and which would compute to a KI of 77.6 micromolar against Mur-E. Next slide. So we extended what we were doing to looking at Mur-C with the same set of compounds. And again, we had to characterize the kinetics of Mur-C with respect to ATP. And at low concentrations, below 0.5 millimolar, the system behaves as a perfectly ordinary Michaelis-Menten saturation, hyperbolic saturation with a Km and a K-cap. If you increase the concentration of ATP, you go into substrate inhibition. And this becomes interesting because this suggests that there's an additional ATP binding site. In other, or that the ATP can bind in two ways. Either way, there's more than one interaction. At the lower end, below 500 micromolar, because you could fit the data to the Michaelis-Menten equation, as well as an equation that describes substrate inhibition, and there was very little difference between the two, we basically elected to drop the concentration of ATP on the basis of the Km of the lower ATP data to 60 micromolar, and then we screened Becker's hits with at that concentration of ATP with UDP Mernac. And this is what we found. Next slide. Next, yeah. So we found numerous hits against Mur-C. We found compounds that targeted C and D, so FO9, C and E, MO2, C and D and E, LO6. So at the moment, we're at the stage of actually more thoroughly characterizing those particular hits against the Pseudomonas enzyme. So next slide, please. Enter our two erstwhile final year project students. So what we did with them, we basically gave them the E. coli Mur-D and Mur-E and asked them to see what they could find with the compounds that we'd already started to characterize. And again, they start with the kinetics, and E. coli Mur-E is again hyperbolically dependent on ATP. As is E. coli Mur-D, again below 500 micromolar, but above that concentration, the enzyme again goes into substrate inhibition, which you can see in the bottom right-hand graph. Again, I'm going to be coming back to that at the end. But what it did mean is that if we worked at low ATP concentrations, we could probably the substrate inhibition that we saw there. But I don't want you to forget that because I'll be coming back to it. Next slide, please. So essentially, these are the IC50 data for E. coli Mur-E and E. coli Mur-D. On each panel, the IC50s run at 2 ATP concentrations, 100 micromolar and 20 micromolar. And so a competitive inhibitor, you would expect to show an IC50 which increased with the ATP concentration. And with JO6, which is on the right-hand side, that appears to be true for Mur-E, and it appears to be true for Mur-D. It also appears to be true for E. coli Mur-D with LO6. So the conclusion, the strict conclusion, is that the binding of JO6 to both E and D is mutually exclusive with the binding of ATP. The binding of E. coli Mur D to LO6 is similarly mutually exclusive. With E. coli Mur E, there was insufficient difference between the two IC50s to really make it a diagnosis. It could easily be non-competitive inhibition. But on first pass, it does seem that the general idea that you've generated an ATP-directed inhibitor in silico appears to bear out in vitro. So we're quite happy with that. Can I go to the next slide, please? So I've talked about the fact there may be two ATP binding sites. But the one thing that is true about all Mur ligases is there are two nucleotide sites. One site is basically the ATP binding site. The other, of course, is the binding of the UDP moiety of the peptidoglycan substrate. So a naive question might be, if we're seeing substrate inhibition, maybe we're seeing interaction at both sites. And if we are seeing interaction at both sites, if we were to actually employ a molecule which had a nucleotide at either end, what would that do? So we gave E. coli Mur E a molecule AP4A, which essentially is a molecule of adenosine followed by 4 phosphates followed by adenosine. So you can think about it as ADP linked to ADP. And it turned out to be an inhibitor of E. coli Mur E with an IC50 of 1.1 millimolar. The kinetics look sigmoid, which may well be consistent with the fact that there are essentially two binding interactions. But the interesting thing is you can actually commercially buy uridine tetraphosphoadenosine. So we're in the process of purchasing that to look at the impact of that molecule, because it might just be that it could be possible to generate inhibitors which actually cross both sites. Of course, that's conjecture at the moment, but it is one of the more interesting spin-offs from our final year project students. So the last thing that we got them to do was we got them to basically trial compound J06 and LO6 in straightforward low volume 10 microliter MICs. And essentially the take home from all of this is that wild type E. coli, wild type Strep galactii, and any strain of Pseudomonas aeruginosa we tried did not have an MIC below 2.5 millimolar. However, a Tol-C minus mutant of E. coli does have an MIC of around about 104 micrograms per mil, which is not outstanding. So an ampicillin control for the equivalent strain will be less than 0.025 micrograms per mil, but nevertheless it's there. The compound is not bacteriostatic. You can plate out the MIC and you get growth in all wells, suggesting that we haven't killed anything, we've just stopped it growing. One thing that does come to mind in all of this is what the compound actually has to do. Not only has it got to get through two membranes, it gets into cytoplasm and it faces an ATP concentration of the order of about 10 millimolar, depending upon growth conditions. So the long and short of it is that it's probably quite a big ask for a molecule, for a fragment with a relatively low affinity to actually be able to do anything under those circumstances. And of course, that's what we're here for, to try and actually swing that balance. And that's that.
Lovely. Thank you, Adrian. Does anyone have any questions?
Wonderful work.
Thank you.
Quick question, which of the compounds or compound derivatives are the ones that have been ordered? I think Laura was mentioning it, or Joe was mentioning it.
As far as I know, we've ordered from Warwick, J06. There was some talk from Northeastern about ordering something, but I don't know if anything has.
Yeah, so those two have been ordered.
Yeah.
Are there, have you done a scan for the nearest available chemical space of enamine?
No, not yet.
Okay, let us know if we
But
can
that's
help with
something to
that.
do.
Happy to see what's available, but that's great.
I mean, we did have sort of internal discussions about what would happen if we trialed some of these things with an inhibitor F1, F1 ATPase, for example, and stuff like this to try and swing the balance, as it were. That's a possibility. I mean, the other thing that we of course have to do is see whether we're actually accumulating any peptidoglycan precursors in those cells as well, which would give us a really strong indication that we were actually targeting the right thing.
I mean, the other thing that's been going through my mind is that Adrian mentioned it. That the one experiment we've done showed these to be bacteriostatic rather than bactericidal. D-cycloserine is variably bacteriostatic or bactericidal, depending on the concentration you're using. On the other hand, phosphomycin at the front end is bactericidal. So I was going to have a chat to Laura about the structures and if we might even be able to think about making conjugates. Is there a possibility to make a phosphomycin Mur C inhibitor conjugate? Because phosphomycin is great for getting in permeabilizing all sorts of cells, gets all over the place.
I can check it out.
Yeah. It's a very small molecule, and it might not. Maybe you can't tinker with it.
Is it known what in the molecule is responsible for its abilities?
There's a resistance mechanism that modifies C1, and that takes out the activity. So we know what inactivates it. Yeah.
It's a covalent inhibitor.
Yeah.
It doesn't hit. I think it hits Mur A, right?
Mm-hmm.
Yeah. So the resistance mechanism in things like TB and mycobacteria is the substitution of the active site cysteine, which is modified by phosphomycin within aspartate.
So I'm assuming, Laura, you're trying to soak these compounds, right? You're trying to get structures with them.
No, I can. I have already apocrystals, so I can soak. I have many apocrystals now. We can soak into them.
Okay, very exciting.
Yeah, it's good. Adrian, did you want to say something about why multi-targeting is probably essential given the concentration of ATP?
Given the concentration of ATP.
Yeah, given the intracellular concentration of ATP, why you were saying why it's probably going to be essential to multi-target.
Oh, I see what you mean, to try and tighten down the ATP concentration.
Yes.
Yeah, I mean, so if we've got an ATP-directed inhibitor that's just about doing it, then its actual impact as an antimicrobial in any sense is going to be dependent upon the physiological state of the cells, which is going to dictate the ATP concentrations within the cells. And if you look at people's various estimates of that, that can be anywhere between 1 to 10 millimolar. Now, the thing is that we've targeted Strep allegalactii, which is a facultative anaerobe, Pseudomonas aeruginosa, which is an obligate aerobe, and E. coli, which is facultative. And so, depending upon the growth conditions, it's not really reasonable to expect the ATP concentration in cells to be the same. So we were thinking that one way to ensure that we might actually swing the balance is if we were to start investigating whether ATP inhibitors, which are actually some of which are currently targeting mycobacterial ATP synthases and are actually in clinical trials, whether molecules like those would actually have a significant impact upon the affinity or the ability of the compounds we're developing to actually target the Mur Ligase pathway.
So again, thinking about, well, we've done this in the past, as Adrian reminded me, about 15 years ago with the pneumococcal Mur C inhibitor.
Yeah, we did. Yes.
Opticin is an ATP synthase inhibitor related to quinine, hydroquinine, and that dropped the MIC back fourfold, didn't it, against this inhibitor?
Yeah. That's right, yeah.
So for TB, I think it's an interesting proposition, given that the development that already exists with the inhibitors, and again being mindful, probably to maintain the multi-targeting, we're probably going to have to retain quite a small size of molecule, and thinking about again chemical conjugation possibly with some of these alternative inhibitors at an early point. So just structurally as we're going through that, and chemically just being mindful, I think, in addition just to pushing up potency and whole cell activity, perhaps thinking if there are opportunities for conjugation as well during the chemical progression.
So what's the difference between ATP concentrations in a bacteria and an ATP concentrations in human cells? I mean, as you know, there are more than 20, 30, 40 kinase-directed, ATP-dependent, competitive oncology drugs. I mean, this was the argument that was made 25, 30 years ago, that you can never have a kinase inhibitor because the ATP concentration was too high. So what's different about these bacterial cells in terms of ATP concentration and human cell ATP concentration?
I think the answer is absolutely nothing. The point really is that it's they both will have millimolar concentrations of ATP. What it comes down to is the ability to generate selective and potent molecules, and the more tightly and specifically binding those molecules are, the more likely you're going to be able to get good competition. The types of tight binding inhibitor that have sort of nanomolar type affinities bind so tightly that the likelihood is that even with millimolar concentrations of ATP swimming around, there is little chance that you get the competition that would actually cause the inhibitor to fail. The one thing that did occur to me about thinking about these things is that some of the Mur ligase inhibitors that we've looked at before appeared not to bind within the ATP binding site, appear to actually bind towards the edge of it, in a non-competitive manner. It does seem to me that although ATP binding site targeting is nice because it's a conserved motif throughout the Mur ligases, if the ATP concentration is going to be something that's a significant factor, it may not be a good idea to forget about the fact that those molecules are there, because that does get around a problem. I think the only issue there is how conserved that binding site actually is, that alternative binding site, because you're dependent upon that conservation for multi-targeting.
So just as a reminder, there's a number of slides on GitHub that compare Mur ligase versus some known human kinases, and there's a difference in how ATP binds between the Mur ligases and human kinases, and our designs have been focused on that difference.
Yeah.
If you also would look at the AstraZeneca Mur-C paper, where they compared their AZ Mur-C inhibitors against a panel of human kinases, you will see that there was selectivity. So I think there's some established precedents here about having selective at least Mur-C inhibitors, and given the conservation between Mur-C and D, for example, I would expect that you can get selectivity between Mur-C/D ligase inhibitor, ATP competitive inhibitors versus human. You're always going to have some kind of issue with selectivity. I mean, this is the bane of so much of the human kinase oncology inflammation work. But people have spent a lot of time and have been able to develop selective human kinase inhibitors. And so I think this is still the best strategy in terms of going after a Mur ligase as an antibacterial target, is to go after the ATP site.
everything I've just said is not a reason for not doing that at all. It's more to do, it's just that because we're dealing with fragments, some of which are actually for fragments quite potent, you know, with KIs of double-digit micromolar. Despite it, I guess what it kind of shows is how much lower we actually have to go to actually get decent, effective competition within the actual cell.
Absolutely. I mean, I think that's the focus. It's going to have to be is driving the potency down. Again, you know, if you look at the DayZ, Mur work we did, I can't remember exactly the initial hits, but you know, yeah, we drove down to those compounds were driven down by 1,000-fold in potency, and that's, you know, hopefully as we, you know, move forward and we get structures, I mean, that's going to be the whole goal, is to drive the potency down, hopefully using some structure and medicinal chemistry effort. So I totally agree. I mean, I think, you know, looking at, I mean, I think it's, you know, nice for your students to be able to, you know, the experiments you showed with the microbiology. I mean, that's you know, nice work. But you know, obviously we need to, you know, drive the potency before we would expect to see any significant MICs.
Yeah.
Anyway, that's great. I mean, it's good. I mean, it's all good. All good.
We've got dead cells. It's definitely good. It's definitely good. So, yeah.
Yeah, it's a good starting point.
Yeah, something to work with.
I mean, I do have a question. I guess I'll put out there. I mean, so I know you know, Becca selected out about whatever it was, around 20 compounds out of the original Mur screen, but there were more hits that were potent in that screen, and I was just wondering, Adrian, you know, where's the status of actually profiling some of those more potent hits that were less than 35% remaining activity. It wasn't quite clear where that status is of that, of looking at the rest of those compounds from the original screen in terms of the you know, further profiling against either E. coli or Pseudomonas Mur Ligases.
So at the moment we're working with a cutoff of about 30%. So we've, with regard to turning around the entire library against the CD, ENF, which is something that we want to do. At the moment, we're still establishing that the stopped assay that we've got is up to the job. The continuous assay that we do use is, but that does have a significant impact upon the resources required to run it. And if that's what we have to do, that's what we have to do. One of the steps that we've taken to cut down the work, because in actual fact, most of the work here is not actually in the lab. It's what you do with the Excel data afterwards that takes the time. So we've got another one of our lab members who's got a bit more expertise in actually data reduction than we have, to actually come up with a MATLAB routine for reducing progress curves to basically a set of numbers which we can far more readily interpret. So that is going to help a lot. So at the moment, Joe, it's a work in progress, but we will get there.
Okay, I mean, thanks, Adrian. I mean, yeah, obviously, I mean, you guys have done a lot. I mean, I'm not, I'm greatly appreciative of finally getting these kind of enamine compound profile. I just didn't want to miss out, because again, I think there were some compounds that looked in that the original MUR-D, Becca's original MUR-D assay, that looked quite interesting below 30%, that for whatever reason weren't selected. And I guess I was trying to understand, why they haven't been further profiled. But like you said, you'll get there. And at some point, ultimately the rubber hits the road in terms of chemistry of what, series, which compounds, which hits are the most attractive in terms of trying to drive a medicinal chemistry program. And so that's the other factor that ultimately comes down to, if you kind of look at a set of hits, where do you want to go in terms of chemistry? What's the most tractable chemistry to kind of follow up on? And so I think having as much of a full set of interest of the compounds of the hits to kind of have a discussion about where we want to go in terms of chemistry efforts, whether that's with the NIAID or a grant proposal, or in Matt's lab, wherever. It's good to have the kind of the full set in hand. That's all.
Yeah, we need extra pairs of hands. Adrian's done nothing but Mur Ligase work for, I can't remember when. Ditto other members of the lab. So I've got two more project students starting for work over the summer. So hopefully they'll be as good as the previous two we've just had, and they can get them to crank through things. But to be honest, pausing and investing the time and effort in getting the stopped assay done would progress an awful lot of things a lot faster.
It would. They really would.
Yeah.
Yeah.
I'm sorry. I'm acutely aware that there's a few minutes left, and I think Jan has an update that we would all like to hear.
Oh yeah, that's true.
Thanks, Laurie.
Yeah, sorry.
No worries. I can make this really quick. So after the last meeting, Laura sent me a UMA compound, and we set up trays. Got crystals for both Acinetobacter MUR-D and Pseudomonas MUR-D. Got data last week. Curiously enough, the Acinetobacter structure was in the APO conformation and didn't contain any UMA. I need to follow up on whether that was human error, that if the UMA wasn't added by mistake. But the good news is that I have now a structure. Share this. So we got 1.6 angstrom density, or 1.6 angstrom structure of Pseudomonas MUR-D bound to UMA. What is very curious here, we got another molecule sitting over here that looks a lot like glutamate. Isn't quite glutamate yet, but it sits there where one of the inhibitors that contain a glutamate has its glutamate moiety sitting. What that molecule is, I'm not quite sure about, but I modeled it at this point as a succinic acid. Glutamate wouldn't quite fit. And then when you overlay this structure here, so we got three structures from Pseudomonas superimposed on this middle domain here, residues 100 through 300. In blue, we got the APO structure from Pseudomonas. In gold or yellowish, we got the ADP bound structure. You see the ADP sitting up here. And in whatever, salmon or coral, we have the UMA bound structure. And you can see that this C-terminal domain swings even closer in the UMA bound structure. So that's hot off the press, got the data last Thursday, and the structure has been in peer review since yesterday, our internal peer review. Once that is done, I will share that structure with the team. And that's all I got.
Thank you, Jan.
So any chances just to zoom in on the ATP? I was just curious, like, if you look how highly similar actually the UMA structure is with the ADP bound structure.
So
you turn off the APO one. Yeah.
So it depends on what you superimpose. I superimpose here on the left side. That looks highly conserved, and here you can already see the domain movement of that C-terminal domain. But I would assume within the week you will have that structure available to take a really close look yourself.
So, so this is Pseudomonas MUR-D, right? Does it make any sense if we sent you if any compounds for any of these fragments were sent to you? Does it? I mean, given your timeline, is there time for you to do any work with Pseudomonas MUR-D, or you're pretty well kind of tapped out at this point?
Yes and no. So in general, I could do that, but the problem is I don't have a lot of protein left.
Okay.
I've been requesting the last bit of protein from Seattle Children's because that's where the protein was made, and that's where the majority of the compounds are, majority of the protein is stored.
Mm.
But their stocks were really low too.
Okay.
I don't know if there's really strong interest. I can ask Bart if he can push another expression and purification, but I don't know how strong their appetite for that is and how many hours they have left they can spend on this. But these proteins crystallize within like a week. So once I have protein, I could move ahead pretty quick.
So Jan, I would say that, you know, we're kind of hurting for protein purification right now because of personnel turnover. But we can grow, we can do our upskills. In fact, we've got a lot of capacity right now. So I would say we should submit upskills to Logan.
Okay. Yeah. Okay, let's talk about that after the meeting.
Okay.
Cool.
Yeah, I'm just wondering, you know, I mean, we have E. coli and MUR-D data that Adrian presented, excuse me, that Adrian presented. I'm just wondering, again, I guess my bias is again a little bit around, you know, the MUR-C, MUR-D, what do you call those? Not dual inhibitors, multi inhibitors. It's the right terminology, I guess. So it'd be interesting if, you know, a couple of the fragments to try to get a Pseudomonas MUR-D structure. But anyway, we can talk about it offline. I know it's the hours late.
Yeah. And I actually have to dash for a company meeting.
Yeah, I think
Yeah.
it's time.
Yeah.
Yeah, well, thank you, everyone.
Okay.
Yeah.
Okay.
Thanks, everyone.
Thank you.
Really nice
Bye-bye.
meeting everyone.
Bye.
Thank you.
Bye-bye.
Bye.